Good morning, everybody. I'm Andrea Ruda from The Ohio State University, and I'll be chairing this session. It's my pleasure to introduce our first speaker, Dr. Jay Calvert from Zoetis. He completed his PhD in Purdue in genetics in 1981. He also completed a postdoc with the USDA from the years 1990 to 1994. And he's been with Zoetis and legacy companies since then, just about completing about 30 years of service. So with that, we'll welcome Dr. Calvert.
Good morning, everyone. Welcome back from the break. Part of me wishes I was watching Glenn and Lucina's talk in the other room, but for you PRRS fans that came here, I'm glad you did. So I'm Jay Calvert from Zoetis. I want to acknowledge my co-authors, Jose Angula and Alvaro Aldez. They're veterinarians. I have to apologize, I'm not a veterinarian. I'm a virologist for 40 years and about 30 years working on PRRS virus. And I'm kind of a gene geek, so recombination excites me. I want to talk today about recombination in PRRS and how we can control it to some extent and eliminate it. Okay. If that doesn't, oh, there it is, sorry. Go back. So the agenda, we're gonna divide the talk up into three parts. The first one is how do PRRS viruses recombine? Examples of recombination between these viruses and strategies for preventing recombination in the field. And I want to start by posing a question and suggest an answer that's important. We've been plagued with new strains of PRRS arriving all the time since the beginning that are different from what's out there, an ever-growing diversity of PRRS viruses. And the question is, where do these new strains come from? And we thought we knew most of the answer because the virus mutates very fast and can add additional amino acid changes here and there throughout the genome and evolve at a high rate, as fast as any other virus. It's right up there with influenza and HIV in terms of rate of evolution. But it's more than just point mutations, right? It's recombination. And we're seeing that now because we're able to see it. We're sequencing more whole genomes, especially in China, where they do whole genome sequencing on almost everything and release it publicly and then talk about it in a paper. There's all kind of recombination going out, not there, but in Europe and in the US. So recombination is a major source of new strains. We can't control mutation, but there are things we can do to control recombination. So let me kind of define the terms here. Recombination has general and specific meanings. In a general sense, recombination is the process of combining genetic materials from two different genomes into a third genome during reproduction. Recombination is a mandatory part of reproduction in pigs and people and most other higher organisms, but not so in bacteria and viruses. It's not mandatory. They're quite capable of making exact copies of their genome with mutations. But in the long term, recombination of whole sections of genomes seems to be essential for the survival and evolution in all species, including viruses. Why is that? Well, recombination is the most effective way to get rare beneficial mutations, different mutations that are in two different genomes into the same genome so you can get the benefit of both of those in one organism. And the flip side of that is recombination is also very efficient at removing deleterious mutations from a genome that's otherwise quite fit. The deleterious mutations from the point of view of the virus include those attenuating mutations that go into a vaccine to make a modified live non-virulent. But the virus would love to get rid of those, so what better way than to swap out that stretch of RNA that has those mutations with a stretch from a field strain and get rid of that mutation. So we're focusing on molecular recombination, not the broad sense, or intramolecular recombination, I guess I should say. We're not talking about reassorting like in influenza. Anytime organisms have a segmented genomes, like chromosomes in people, or segmented genomes like in influenza or rotavirus, you can have reassortance, and that's where whole segments go here or they go there and they reassort. PRRS is a single strain of RNA, so its genome is just one molecule, so reassortance is not an issue in PRRS. Instead, we're talking about recombination and swapping of segments within a single strand. Right? And it can happen in DNA and RNA to create a chimeric hybrid virus. But the mechanism, especially in single-stranded RNA viruses like PRRS, is very different from the mechanism in a double-stranded DNA genome. So let's go over that in a little diagram here. Kind of busy, but it's not that bad. Here's two genomes, two PRRS genomes, in, we'll call that blue and that's purple. They find themselves in the same cell of a pig at the same time, which is not something that you would normally see, but you certainly can see it if you vaccinate and then have an outbreak, or if you have a double outbreak, or if you use two vaccines at the same time. You can have two PRRS genomes in the same cell. When that happens, let me go back here. When that happens, as this purple strand here, this is a positive strand, this is the genome, it's making a negative strand. Here's the polymerase moving in this direction. It's making a nascent RNA, that's the brown bit here. At some point, more or less random, not really random, but many places throughout the genome, this can disconnect. It can float away, and it looks for another template to bind to. Maybe it binds back to the same one, or one that's very similar from the same infection. But if there's two virus genome types in there, it could also bind here if there's sufficient homology. And from there, continue to transcribe a negative strand, like this. And you end up with this. This is a negative strand, and from here you make new positive strands. And you can see this is chimeric from two different genomes, and the same with the transcribed positive strand. This gets packaged into virions, and from there it can infect other cells in the pig and other pigs in the herd, and even other herds nearby. So that's the mechanism. A couple of questions. Is recombination in PRRS a new problem? It's not a new problem. It's been there from the beginning. But back when the diversity of PRRS was low, you didn't notice it because it was just another new strain. And it was a little bit like the existing strains. With such a high diversity now, including some very highly virulent viruses out there, when you get a recombinant, it can get noticed. Combined with the whole genome sequencing, it's easy to find out and ask the question, is this a recombinant virus? And so it's not a new problem, but it's a bigger issue now than it used to be. Okay. Are all the recombinant viruses virulent? Absolutely not. Most of them are not as fit as either parent because they're taking proteins with amino acid sequences that are from different viruses and asking them to interact with each other, and there's going to be some poor matching and misfits. So most of the recombinants are not going to be more fit than their parents, but some will be from time to time. And if they give them a fitness advantage, for example, if they can replicate faster than either parent, they will outcompete the parents. They'll appear at a level that's detectable by sequencing. They'll grow probably faster if they're more fit. They'll grow faster than the parents. And these are the ones that tend to get noticed, and they go to diagnostic labs because there's some clinical signs, and they get sequenced, and you can see how that happens. Some examples, quickly. I'm going to give you 3 of them. This first one is from the US from a few years ago. It was in a farm that was now experiencing some abortions in sows after using 2 different commercial vaccines. Problem is they used them at the same time. So I think they were in the process of switching, and they used the old one until they ran out and then kept vaccinating at the same time with the new one. And so we have at that, basically a worst case scenario for recombination between vaccine strains. They're both vaccinated in the same farm at the same time. And this is what was recovered from that. This is a recombination oops, I keep hitting the wrong button. Recombination analysis. The way to read this is the genome from this side to this side with the bases there. We get 99% similarity if we read across the top, but it alternates from vaccine B to A to B to A to B, and so forth. There's 8 different segments. Each of those 8 segments is at least 99% similar to one virus or the other of the 2 vaccine strains. And since the vaccines are pretty far apart, so if you look at these numbers, 94 down here around 90%. So these are 10% different. So it's easy to tell which one is similar to either A or B. Yeah. So to have 8 different segments, it means there were 7 individual, probably sequential, recombination events. So the virus really got down to business fast, with both vaccines hitting peak viremia at the same time. And what it's doing is it's throwing out the segments that contain the attenuating mutations from the 2 different commercial vaccines. So the mutations in vaccine and the mutations in vaccine B that attenuate it are in random places throughout the genome. And the virus is figuring it out by getting rid of those pieces that contain the most important attenuating mutations. And so you get an increase in virulence, an increase in growth, and the ability to replicate and spread with the recombinant. This is what we're trying to avoid. Okay. Wrong way. Second example, this is from Europe. This is type 1 PRRS from France. And briefly, you can read this one the same way as the other. Read across the top, you see blue and red alternating. In this case, there's 7 segments, so there were 6 recombination events between these 2 vaccine commercial vaccine viruses. The interesting thing here is this has been around for a while, several papers published on it. At first, there were no clinical signs, but some interesting growth changes, growth characteristics, and that's shown on the right side. So this is showing when is the first day of viremia in the pig after infection. And the 2 vaccines are up here. And you can see that these vaccines have a very late appearance of vaccine viremia after vaccination. It's 14 days in the one and 21 days in the other. But in the same study, they put the chimeric recombinant in there by day 2 after infection. Everything was infected at the same titer. By day 2, they were seeing the virus in the serum already, and a lot of it. If we look at the growth curves, not only is the replication rate faster for the recombinant, it reaches a higher peak titer, and it stays at that higher peak titer longer. So that would explain its fitness benefit, and why, last time I looked, it was still circulating in France. And I'm not sure if it's causing some clinical signs now. It might be. I have to follow up with that. And the third example is also from Europe in Denmark. This is PRRS1. This is the so-called Horsens virus. Some of you may have read about it. But it was a recombinant between 2 vaccine strains. And again, we know the history here. This is a new user that stopped using his old vaccine, and after a short period of time, I think a week or 2, he started using the new vaccine. So they were close enough together. But this one was caught much earlier in the process than the last one. There's only 4 vaccine segments, so 3 recombination events. There's also, interestingly, there's this. So it might have 3 parents because there's a short segment there of a little over 100 nucleotides that don't resemble either vaccine parent, but they do resemble a Danish field strain. So it may be recombination between a field strain and the vaccine and another vaccine. Strategies. Okay. Let's look at the vaccine labels, but first let's look at the second line here. You'll see that a couple of times in the presentation. The bottom line from this, if you take anything away, is try to avoid infection of the same pigs with 2 different PRRS viruses at the same time. And that means vaccines, field strains, or combinations of them. You can't avoid that happening sometimes, but anything you can do to reduce the occurrence, especially of high titers, of a vaccine virus and a field strain, 2 field strains, 2 vaccine viruses, try to do that. Because we don't want these new strains arising that are recombinants. If nothing else, even if they're not virulent, they avoid classification and detection. And you get something that looks like a vaccine virus based on OR5 sequence, but it's 90% a field strain. And that gets sorted out eventually, but only through whole genome sequencing. So the common language in the US, they don't have specific words about recombination risk yet, but they all have this word healthy. Use this in healthy swine. And I would remind everybody that during an active PRRS disease outbreak, your pigs are not healthy by definition. So you wouldn't want to vaccinate then. For one thing, the vaccine won't do its job right. It won't be able to replicate well in the presence of a more aggressive virulent field strain. And so that'd just be wasted vaccine. But more importantly, for this purpose, it could lead to recombination. So what can you do? You can do several things. I mean, if the timing is such that you wish you could vaccinate now, but you really shouldn't because of an outbreak, you can wait. How long do you wait? I mean, if you wait till the clinical signs are over, that probably means that many of the pigs that had the field virus have cleared the virus, and the ones that haven't will have a much lower titer. The virus is kind of on its way out. Maybe that's a few weeks, maybe it's a few months. But then you can do the whole herd vaccination. Also, whatever you do, you might want to consider closing your herd and/or increasing biosecurity, because the last thing you want when you have an outbreak is to have another outbreak with a different field strain for a number of reasons. But one of them is you don't want to get recombinations between those 2 field strains. In Europe, it's more clear. They have language on every vaccine now that's specific to avoiding recombination. This is fairly recent. Part of it is a consequence of the Horsens virus I showed you. They're all pretty similar. Let's focus on the second one down here. Let's look at porcellus. 3 different points they're making here. I'll just read them here. Do not routinely rotate 2 or more commercial PRRS modified live vaccines based on different strains in a herd. So this is a practice, some places, to intentionally buy different PRRS vaccines, modified lives, and you use vaccine A, and then next time you use B, and then maybe you use C, or you go back to A, and you rotate the vaccine thinking you're going to get broader immunity. Don't do that. You won't get the immunity you want, probably, and you're going to have a horrible recombination situation. The second point here. Do not use different PRRS-modified live vaccines based on different strains of the same genotype on the same farm at the same time. A lot of difference and sames in that sentence. But I think that makes sense. When they say the same genotype, they mean what they mean. We would call it the same species now. PRRS1, which used to be European PRRS, PRRS2, which is North American PRRS. Those are now different viruses, different species. Within PRRS1, you get a lot of recombination. Within PRRS2, you get a lot of recombination. Nobody has ever seen recombination between PRRS1 and PRRS2 in nature. It's kind of good news. It also justifies the classification as different species. But that's what they're referring to when they say, of the same genotype here. And then transitioning from one PRRS vaccine to another. A transition period should be respected between the last administration of the current vaccine and the first administration of the new vaccine. And that period should be close to or equivalent to the time it takes for the old vaccine to disappear from the herd. But the more time, the better in that case. A cooling off period is what I would call it. Okay. So I'll move on from there. We did that already. All right. So we're getting to the summarizing bit. Basically, and these are rules. I'm going to say do this, don't do that, but I'm not the veterinarian. You guys know what to do. I want you to consider this information, though, when making decisions and management of the herd. If you want to avoid recombination, first of all, do not infect pigs with two different PRRS at the same time. If you have an outbreak, you can delay whole herd vaccination. If you're not using whole herd vaccination, you might want to consider doing it because it makes management of recombination easier. And don't use more than one modified live at the same time in the same location. So, use the same vaccine on the sows and the incoming gilts, right? And you want to vaccinate the incoming gilts before they get into the sow farm with the same vaccine that is being used in the sow farm, right? So there's a lot of different possible combinations. Maybe your gilts got a different vaccine when they first went into the gilt developer unit, but here they are now getting ready to go into the sow herd. You don't want to bring that gilt in like that. First, they may not have strong immunity, and they may still have a little bit of other vaccines. So give them another shot of the vaccine the sow herd has before the gilts go into the sow herd. Use the same vaccine in the sows and the piglets. If you don't, especially if you're doing pre-weaning pigs, so you're using a vaccine where you can vaccinate at processing, you can have a two-way shed of vaccine and have both the sow and the piglets become reaction vessels for recombination. When the pigs leave the nursery, if they're don't mix pigs in the nursery that have been vaccinated with different vaccines. Maybe they came from different sites, or maybe you're in the process of switching vaccines. You should keep them separate for a while until the vaccine titers go down. And let's go to this, maybe one of the last slides. Switching. If you need to switch from one PRRS vaccine to another, do it with a lot of thought and preparation and for good reason, and don't do it often, because you have to modify your protocols a little bit to prevent recombination. So after a final whole herd vaccination with the old vaccine, probably completely discontinue use of that vaccine on that site, and maybe even remove the bottles so nobody's confused or tempted. Then if you time it right, you wait till your next whole herd vaccination, which might be 3 or 4 months away, before you give the first dose of the new vaccine to the sows and the replacement gilts. I'd say at a bare minimum wait 5 weeks, though. Depends on the timing. But if you plan ahead and you're doing whole herd vaccination, you've got a 3 or 4 month interval that could be your cooling off period. Okay, the vaccination of the piglets during this cooling off period may be problematic depending on how you're doing it. If you're normally weaning on site before they leave, the piglets leave, maybe don't do that. Vaccinate them on the way to the other site or when they arrive at the other site with whatever vaccine the other site is using. If you can't use the same vaccine on the piglets. Yeah. And again, about the dedicated nursery, I mean, if you're changing from one vaccine to another, but you're weaning pigs every week, where are you going to put them in the nursery? And if you have a separate nursery temporarily, where you can put the new batch of pigs that were just vaccinated with the new vaccine and keep them away for a period of time from the pigs that are a week older and have got the old vaccine, eventually they can be brought together again when the vaccine titers go down. Maybe when they leave the nursery. And the last bit, I talked a little bit about that. A cooling off period is good. And I'll finish up here. I think I'm running low on time. So again, new strains are bad and they're happening all the time, now more than ever. They come from two sources. Mutation we can't do anything about, but recombination we can manage by just keeping the pigs as much as possible from being infected with two different PRRS viruses at the same time. And I think that's it for me. Thanks for your attention.
All right. What questions do we have for Dr. Calvert? We have our volunteers with microphones, so just raise your hand. If I can't see you, they can. There you go.
Right. I have a quick question. Has to do with the viruses. What is the frequency of recombination that you typically see in situations when you have more than one vaccine being given in a herd? And second part of that is then, how often are those recombinants more fit than the prevailing viruses that are in the population?
I think we don't have enough information to give numbers, but I think the answer to the first question is recombination is much more prevalent than we think it is. But the answer to the second question is the more virulent. I mean, more fit, yeah. I think more fit, it only takes one recombination event. But actually increased virulence, I think that's a rare recombinant, often several rounds of recombination before you get something that's going to cause more disease most of the time.